Hello everybody! In the next 25 minutes, I want to tell you what I did in the last years for my PhD. What I can tell you already now is that I feel like I am a little bit closer to understanding what happens in our brains when we see other people in pain and what prompts us to help these individuals. Doing a PhD at the SCAN unit meant that I was able to combine my psychological background with a social, cognitive, and affective neuroscience approach. When I started, I was really interested in not only how we experience pain ourselves, but also how this is related to us processing the pain of other people around us, friends, family, even strangers. How are these processes implemented in the brain? On top of that, does our own pain processing and our capability to feel empathy play a role in motivating helping behavior? Especially this intersection here is the core of my PhD work. And to investigate this, I used mainly fMRI, a clever method called placebo analgesia, and lots of pain. But why is investigating how empathy is processed in the brain so important? We are inherently social beings, be it in friendships, at work, or in romantic relationships. We constantly have to process and understand our surroundings. And a vital part of these surroundings are dealing with emotions, both positive and negative, both in ourselves and in other people. Those emotions then play a big role in our motivation and actions. For example, they can make a difference whether we decide to help someone or not. The majority of social neuroscience research has used somatic pain as a model to investigate empathy. Why is this so? Pain can be easily and robustly induced in psychological experiments. And there is already strong evidence regarding the neural networks underlying pain processing. I also want to give you a quick explanation of how pain is defined. It constitutes an unpleasant sensory and affective experience associated with actual or potential tissue damage. As you may have noticed, this definition already includes two major components of pain processing that I want to introduce next. Pain is considered to be a complex, multidimensional experience. If we look at first-hand pain, so pain we experience ourselves, two major components have been identified. The sensory discriminative aspect is a more basic representation of pain. For example, the intensity, the location, the timing, or other physical characteristics. They are represented in the primary and secondary somatosensory cortices, depicted here in green. On the other hand, there is the affective motivational aspect, which describes the subjective judgment of feeling pain, so the unpleasantness and any intentions to withdraw from that pain. This component is represented in anterior and mid-cingulate cortices in red and anterior insula in brown. If we see someone in pain, we can understand and feel that pain in ourselves. For example, if I show you these pictures of my former master students accidentally hurting themselves, some of you might discover an unpleasant feeling in your stomach, or maybe even pain directly in your hand. Of course, previous research defines empathy a bit more systematically and as a process consisting of multiple components. First of all, we have to either directly observe or imagine the emotion of another person. Second, we have to then isomorphically share this affective state of the other person in ourselves. We are feeling with the other. Thirdly, we need to know that the source of our emotion that we're feeling right now lies in the other person and not in ourselves. Empathy can therefore also be seen as a combination of more automatic bottom-up and more controlled top-down mechanisms. In my PhD, I focused on this effect-sharing component and wanted to clarify the relationship between self and other directed emotional experiences and how they influence our behavior. Prosocial or helping behavior can be defined as any voluntary behavior that benefits another person but is costly to ourselves. Many studies posit that prosocial behavior is crucially connected to empathy, although this connection may not necessarily always be very straightforward in the sense that more empathy leads to more prosociality. In contrast, empathy can also induce something called personal distress, which may lead to withdrawal and even aggression. Prosocial behavior can be manifold, and this has been measured in different ways in the past. But I want to make a case here that in everyday life, prosocial actions require motivation put in at one exact moment in time. For example, we need to put in actual effort in order to help someone who has fallen off their bike by crossing the street to help them up and risk being late for an appointment. Previous studies have already investigated these relationships and found overlapping activation in the brain in both components of the pain processing network when we experience pain ourselves and when we observe pain in others. Here you can see that while the green parts are active during first-hand pain alone, the red parts are active in both situations. Causal research also showed that when you give people a pill presented as a strong painkiller, it not only reduces their own pain, but it also dampens how they perceive the pain of others. That works if the pill is actually a painkiller, but also if it is not. This method, called placebo analgesia, led to lower own pain, but also to lower empathy ratings and reduced activity in affective brain regions. These results suggest that so-called shared representations underlie first-hand pain and empathy for pain, meaning that we come to understand and share the pain of others by feeling their emotional state in ourselves as if we were experiencing it firsthand, and using similar brain regions in both cases. So, what are the open questions? What is still unclear? First of all, while evidence for shared representations exists for affective brain regions, such as anterior midcingulate cortex and anterior insula, evidence for somatosensory regions has been less clear. These regions are active when we experience pain and empathy, and this differs depending on what tasks you use to measure empathy. But do we also use these more basic pain representation regions to share the pain of others? And second, if our own pain experience influences our emotional resonance to others' pain, we can go one step further and ask if and how our own pain perception influences our helping behavior shown towards others. These questions formed the core of my PhD thesis and can be summarized in two parts. What role do somatosensory representations of pain play in empathy for pain? And how are shared representations connected to our prosocial behavior? In all three studies, participants received a sham treatment presented as a painkiller from a study doctor. They were then conditioned via verbal suggestions and direct experience to believe that the treatment they received was actually working to reduce their pain. What placebo analgesia does, it downregulates your own pain perception so you feel less pain. We were now interested in how such a situation, so when you cannot process your own pain normally, would affect your social emotions and your social behavior. Here's an overview of my three studies, which I will explain in more detail on the next slides. In study 1, I used a cue-based empathy for pain task. Before, participants had undergone a localized placebo analgesia induction on their right hand only using a cream. This allowed me to compare within subjects how the placebo effect induced for first-hand pain on the right hand only compared to pain on the left hand, and how this affected empathy for pain in the form of electric stimulation to the hand. In study 2, the same localized induction in the same subjects was used to measure picture-based empathy for pain, as this is the second prominent paradigm to measure empathy in the literature. This allowed me to investigate whether the placebo analgesia effect would transfer to perceiving pain of others in everyday situations and without a direct first-hand experience of pain. Finally, in study 3, I used a global placebo induction procedure with a pill in a between subjects design to gauge whether downregulated first-hand pain perception would affect prosocial behavior. Here's a brief summary of the measures that I used. In all three studies, I used subjective ratings of self and other directed pain intensity and unpleasantness to measure empathy. Studies 1 and 2 measured bold activity in specific regions of interest of previous studies. And study 3 included multiple measures of prosociality, choice behavior, reaction time, and exerted physical effort via a grip force device. For study 1 and 2, participants came to the MRI scanner together with another person posing as a second participant. Participants completed a cue-based and a picture-based empathy for pain task after they had undergone a localized placebo induction on their right hand only. What did I find in the first 2 experiments? I successfully induced a first-hand placebo effect in my participants within subjects. That means participants had a high belief in the effectiveness of the medication and significantly increased belief ratings after the conditioning procedure. They also rated the pain that they received in the pain task as less painful on their right hand compared to their left hand, and this belief even lasted until the end of the session. This first-hand effect was also visible in secondary somatosensory cortex, where there was an activity difference for the 2 hands. But in contrast to what we predicted, we did not find a transfer of this effect to our empathy condition. So when participants rated the pain that other people received on their right or left hands, or when people evaluated pictures of other people's hands in pain. Bayesian analysis further provided evidence of absence of a modulation of somatosensory regions, and thus do not support the existence of shared somatosensory representations using our task and design. In conclusion, empathy for pain might rely more on sharing the effective consequences of another person's painful situation, compared to the exact nociceptive source. In other words, we might empathize with the pain someone is feeling generally, not necessarily with the pain someone is feeling in their right hand. Even with blunted first-hand experiences focused on a specific body part, are we able to share the pain of others, possibly using the affective network. This is true both when we just infer the pain and when we directly see it in pictures. Especially in artificial experimental situations like ours, the general processing of someone's pain might be weighted higher than knowing and processing the exact location of that pain. This might be different when the location is more important, for example, in medical professions or emergency situations, where help has to be provided based on the exact bodily origin of pain. Nevertheless, these findings bring us one step closer to understanding how we empathize with others in pain, and they underline the magnitude of influence that our own pain experience has on our ability to feel empathy. So, what about prosocial behavior? Do shared affective representations play a role in whether we decide to help someone or not? Spoiler, they do. This is the session overview for study 3, where participants either received a pill presented as a strong painkiller or no pill, after which they as deciders had to make choices about exerting physical effort or not in order to reduce the number of shocks another alleged participant received. Again, I successfully induced a first-hand placebo effect in my participants, this time between subjects. That means participants in the placebo group had a high belief in the effectiveness of the medication and significantly increased belief ratings after the conditioning procedure. They also rated the pain that they received in the pain task as less painful compared to the control group, and this belief again lasted until the end of the session when I asked them again how the pain was that they remembered. We did not find group differences in how participants perceived the pain of others, but we did find differences in prosocial behavior. In this task, participants had the chance to decide whether they wanted to exert physical effort in order to reduce the number of shocks another participant received live, so trial by trial. While both groups helped equally often when being able to reduce more shocks, as you can see on the left side of the graph, the control group was more motivated than the placebo group to also help in situations of smaller pain reduction, so when they could only reduce the other shocks by 1. This you can see on the right side of the graph. Furthermore, while reaction time was not affected, when deciding to exert effort, the placebo group put less energy in their actions, so they pressed the grip force device less than the control group, independent of the effort and shock levels that we had included in our task. However, in an exploratory mediation analysis relating these three concepts, we found that the effect of placebo analgesia on prosocial choices was fully mediated by the level of affect sharing in response to others' pain. So the level of unpleasantness that people felt when the other person received painful shocks. That means that the placebo effect we had induced affected how prosocial people were, but this effect was dependent on how much empathy they felt for the other person. What could these findings mean? Our own pain processing is crucially connected not only to our ability to empathize with others in pain, but also to our prosocial behavior. As Professor Preston kindly pointed out when reading my thesis, there might be a threshold over which the placebo cannot compete with people's empathic pain, but at minor levels you can see the impact. This is interesting when we think of situations where the social norm or urge to help is very strong, versus when it is more ambiguous. Only in the latter placebo analgesia had an influence, pointing to the conclusion that our own pain perception plays a role for our actions, but only when we're less driven by social norms. This is well in line with the altruistic response model promoting fast, intuitive decisions through reward expectation. In the more ambiguous, lower helping situations, reward is decreased, which is potentially why placebo analgesia may exert its effects stronger. These findings are also unlikely to be explained in terms of a general reduction in reward-seeking motivation, as we found no group differences in our control measures of strength, motivation, or mood. This is in line with Mishkovsky, who also found that acetaminophen did not affect general mood. But these results and this interpretation will have to be confirmed by future studies. We also replicated findings of Lockwood and colleagues, who found superficial prosociality, so participants choosing to help, but then pressing the grip force device less when exerting the chosen effort. Placebo analgesia possibly increased this effect. Finally, while the literature has been debating how empathy and prosocial behavior are connected, our results show that how much pain we feel and how strongly we feel the pain of others influences our motivation and actions. This is immensely important when we think of the many people with chronic pain conditions or people taking painkillers on a regular basis, but also for society as a whole. Of course, my PhD work is not able to answer all of the open questions, some of which I want to mention here, as they also lead to some interesting new avenues for future work. We specifically investigated the role of primary and secondary somatosensory cortex, so sensory discriminative aspects of pain processing. However, this component also includes other brain areas, such as posterior insula. At the same time, prefrontal regions have been shown to be equally important in placebo analgesia responses. Future studies could focus on investigating other associated regions and have a closer look in which concrete situations the processing of such localized pain aspects is more relevant. We also have to consider the possibility that somatosensory representations exist, but only on levels with more impactful experiences, depending on the nature of the demand. Furthermore, using placebo analgesia as in previous studies, we assume to directly manipulate the body's endogenous opioid system, as that system has been shown to be involved in shared affective representations. However, we did not specifically check whether this was the case here and can only indirectly assume it. Pharmacological manipulations using opioid antagonists might shed more light on how placebo analgesia affects especially prosocial behavior. Similarly, research has shown that other neurotransmitter systems, such as the dopaminergic system, are involved as well. We used a dichotomized approach when categorizing responders and non-responders to the placebo manipulation, as in previous studies. However, this might have prohibited us from detecting subtle differences in the magnitude of the placebo response and how these differences relate to our outcome measures. As we have seen in the mediation analysis, measuring placebo analgesia continuously from no response at all to a very strong response might be a more nuanced direction for the future. Moreover, pain is not the only, although a very prominent emotion to focus on when investigating empathy. In order to check whether the here found effects generalize to other emotions or are specific to pain, more studies are needed. Lastly, more work is also needed on what these findings mean for people under the influence of opioids or painkillers in general, as well as people with atypical pain processing. How do these subgroups perceive the pain of others? And how does this influence their actions? How does this relate to using affective versus somatosensory representations? How are the needs for oneself weighted against the needs of others? Comparing brain activity in my used task in these groups to typically developing individuals might shed more light on these aspects. Finally, here are some of the other projects that I was involved in over the course of my PhD. Those studies covered diverse topics such as meta-science and open scholarship, feminist psychology, as well as personal distance, social cognition abilities, and therapeutic approaches in autism spectrum disorder. Many other projects are also already submitted or in preparation. Before I close, I want to draw your attention to two important points that are very dear to my heart. I was one of the first people in the lab to start pre-registering their PhD studies and incorporating open scholarship methods into their work. This was really a deep dive into unknown waters for me, especially pre-registering an fMRI study. Nevertheless, I encourage everyone to start thinking about the transparency and reproducibility of their own work and what they can do to improve it. Second, I was able to use my PhD research to engage in tons of science communication, from written work to podcasts and videos, online as well as in-person events, old to younger target groups. Apart from communicating research to the public, science communication taught me to see the bigger picture and to contextualize my findings better. I wholeheartedly think that science communication makes me a better researcher. Now it's time for some thank yous. A huge thanks goes to Klaus for not only supervising but also mentoring me on this journey, together with Georgia and Wolf as my thesis advisory committee. I also want to thank Stephanie Preston and Dominik Mischkowski for acting as external evaluators of my thesis. Your comments brought up very valuable points that I can incorporate in my work and writing going forward. Both of your research inspired me hugely during my PhD and still does so today. Lastly, I want to thank my lab, the SCAN unit, and also my lab where I went abroad, the Social Brain Lab, for the constant support during sleepless nights with the pain stimulator, all my students who have helped me collecting the data sets, and my funding that made this PhD possible in the first place. It has been an exciting journey for me that I hope will continue in the future. Thank you all for either playing a part in it or listening to this talk today.